All right. Thank you, everyone, for getting up so early to come to this talk and this session. So before I begin, I'd like to acknowledge those who led this work. So the work that I'm going to be talking about today was led by Patrick Sadler and Matthew Golub, PhD students and postdoc with us. And they were co-advised by Professors Aaron Batista and Steve Chase and me. And Matt, Aaron, and Steve and I are all here, so feel free to chat with us afterwards if you have questions. Okay, so when you hear the term brain-computer interfaces, the first thing you probably think of is a paralyzed person controlling a robotic limb. Okay. While we're very interested in this as well, this is not the main focus of today's talk. Okay? And there's a use of brain-computer interfaces that has not received much attention, which we think has great potential. And that is using brain-computer interfaces to address basic scientific questions that are currently difficult to address using other means. Okay, so the central message of today's talk is that brain-computer interfaces represent a powerful tool for addressing basic scientific questions. Okay, what do I mean by this? Well, we can view brain-computer interfaces as a way of simplifying the brain's output interface without hopefully simplifying the complexity of brain processing that we seek to understand. Okay, so let's say a little more about this. So the brain's output interface is complex. Okay, so for example, if we take arm reaching, the effector is the arm, and the number of non-output neurons are in the many millions. Right? These are all the neurons throughout the brain. The number of output neurons, there are many thousands. Right? These are neurons that project down the spinal cord. And in the best of experiments, only some of these are recorded at any given time. The neuron-to-movement mapping is unknown. The effector dynamics, right? So the many degrees of freedom in our arm movements, as well as the nonlinear dynamics of our muscles, right, make things very complicated. And also sensory feedback, in this case, I'm referring to proprioceptive feedback, is tied to the arm. Okay, so all of these things together make sensory motor control with the arm hard to understand. So what we propose here is to use a brain-computer interface to simplify the output interface of the brain. So how a brain-computer interface works is we record neural activity from a population of neurons, and then we map the activity of these neurons to a cursor moving on a computer screen. Okay, so here I've shown an equation that represents what we use. And so here, x sub t is a two-dimensional vector. That's the horizontal and vertical velocity of the cursor. X of t minus 1 is the velocity at the previous time step. U of t is a vector representing the spike counts from each of the neurons that we record from at the same time. Right? So the length of this vector is the number of neurons we record simultaneously. And big A, big B, and little b are the parameters of our BCI mapping. Okay, and then what the subject needs to do is to use visual feedback on a moment-by-moment basis to move a cursor to hit targets on a computer screen. Okay. So in a BCI, our effector is now a computer cursor or a robotic limb. The number of non-output neurons is still in the millions, right? These are all the neurons in the brain, and they all work together during BCI control. A key difference here is that the number of output neurons is smaller, and we get to record from all of them, okay, by definition, right? In the way in which we've set up the brain-computer interface, all neurons in the brain can participate, but only those neurons that we record from can directly affect movement. Right? And so this is going to be extremely powerful for us because we're going to be able to make causal statements about if we see changes in behavior due to learning, for example, we can ask which neurons were responsible for it. Furthermore, the neuron to movement mapping is known because we, as experimenters, get to define what that mapping is. And we can define that mapping to be simple and linear. Okay, so for example, the one that I showed you on the previous slide. And furthermore, sensory feedback is flexibly manipulable. And what I mean by this is that in a BCI, proprioceptive feedback is not tied to where the cursor is. Okay. So I'm going to show you a video, okay, that is typical of very good cursor control used by a monkey with a Utah array implanted in the primary motor cortex. Okay. So what we see here are movements that are very accurate. They're typically 100% correct, and it usually takes about 1 second for the cursor to move from the center to acquire these targets. And by most metrics, this is about half as good as what we can do with our arms. Okay? And the reason why I'm showing you this video is to make the point that a prerequisite for us to do scientific studies with BCI is to have good behavioral control. Okay? And we have that here. Okay, there are several key advantages of using a BCI. The first is that we can monitor all the neurons that directly drive movement. Okay? And so, as I mentioned before, this allows us to make causal statements about when behavior changes, what actually changes in the brain that actually led to those behavioral changes. Second, we can distinguish between output and non-output neurons. Okay? So for example, you can record from, say, 100 neurons and use only a fraction of them for controlling the BCI cursor and passively monitor the other neurons. Right? And so this is relevant for addressing questions involving readout between one brain area and another brain area, because not all neurons in brain area A project directly to brain area B. Right? And so we can address questions about what happens to those neurons that do not directly feed to the downstream area or the readout. Thirdly, we can define a simple mapping, like the one that I showed you on a previous slide. And this is extremely enabling because we can test certain hypotheses with linear mappings that are going to be a little harder to test if we had nonlinear mappings. And I'm going to show you an example of this. Furthermore, we can change the BCI mapping during the experiment. Okay, this is going to be a very useful tool for us because now we can study questions about learning. Right, and I will illustrate this as well. Okay, and then lastly, we can flexibly manipulate the sensory feedback. Okay, so I won't be talking much about this, but there have been studies where you can change the proprioceptive feedback separately from the visual feedback. Okay, so in the work that I'm going to be telling you about today, we're going to leverage advantages 1, 3, and 4. Okay, and the particular question we're going to study is about learning. Okay, so learning requires the brain to alter the neural activity patterns that it produces. Okay, so for example, when you're learning to play tennis or when you're learning to manipulate a Rubik's Cube, something changes in your brain between when you go from a novice to becoming an expert. Okay, so we're going to address 2 key questions. Okay, the first question is, why are some tasks easier to learn than others? Okay, and then the second question is, how does neural activity change during learning? Okay, so why are some tasks easier to learn than others? Okay, so if learning requires the brain to alter the neural activity that it produces, then perhaps it's the case that certain types of activity patterns are easier for the brain to produce than others. We can go even farther to ask, can the brain produce arbitrary patterns of population activity? And if not, what defines the constraints on learning? Okay, so let me show you a cartoon of the experiment that we designed. So let's say we record from a population of neurons. So in our experiment, we recorded typically from many tens of neurons. Okay, but here in this illustration, I'm just showing 3. What we can then do is to define a population activity space where each axis represents the activity of one neuron. We can then take a window in time. We can count the number of spikes that each neuron produced, and that gives us 1 point in this space. We can then take many non-overlapping windows in time, and this gives us many points in this space. Now what we can do is to define a BCI mapping in this space. Okay, for example, we can define this BCI mapping shown here, right, whereby to move the cursor to the left, the subject would need to move its neural activity to this lower left part of the space. Right? And to move the cursor to the right, the animal would need to produce neural activity out here. Okay, the key experimental manipulation that we're going to use here is to change the BCI mapping. Okay? So now, in order to move the cursor to the left, the subject will need to produce neural activity up here. And in order to move the cursor to the right, the subject will need to produce neural activity down here. So right after this switch, the subject's performance with this BCI will be poor. Right? But with trial and error, the subject can learn, right, to perhaps control this BCI mapping. Okay, so this is going to be our central question, right? So when we switch the BCI mapping, can the subject learn to control the cursor using this new mapping? Okay, so this led us to wonder whether there was some principle underlying what types of mapping changes would be easier to learn or harder to learn. Okay, and our core hypothesis relies on an empirical observation that we and other groups have observed, okay, which is that neural activity does not uniformly populate this population activity space. Okay, rather, the neural activity tends to lie within some low-dimensional space, okay, which we call the intrinsic manifold here. So this intrinsic manifold presumably reflects constraints imposed by the underlying neural circuitry. Okay, so for example, if neurons 1 and 2 anti-vary, right, due to the underlying network, then that will be reflected in the relationship here between neurons 1 and 2 in this intrinsic manifold. Okay, so this intrinsic manifold is a direct reflection of the pairwise correlations that lots of us measure here. Okay, so our hypothesis was as follows. Okay, so we start off with a BCI mapping that looks like this. Okay, so this is a BCI mapping that is easy for the animal to control, okay, like in the video that I showed. We're going to introduce 2 different types of mappings. Okay, the first type of change mapping we call a within-manifold perturbation. Okay? So this within-manifold BCI mapping, okay, requires the subject to use neural activity patterns within the intrinsic manifold, but now for different movements. Okay? The second type of switch in mapping is an outside manifold perturbation. Okay? And this type of perturbation encourages subjects to show neural activity outside of the intrinsic manifold. Okay. So what we hypothesized was that it would be easier for the subject to learn within the intrinsic manifold compared to outside the intrinsic manifold. Okay. So before someone raises their hand and says, isn't this obvious? Like, why are you even testing this? Right? I want to put two things on the table. Okay. The first is that right after the switch in mapping, both types of mappings were equally difficult. Okay? So it's not the case that within manifold mappings were initially easy and outside manifold mappings were initially difficult. Right? So what we do is we switch the mapping, performance goes down, and then we watch to see, through learning, how performance recovers. The second thing I'd like to put on the table is that other studies have shown that subjects can learn outside manifold mappings. Right? So it's not as if these outside manifold mappings are impossible. Okay, so let me tell you a little bit more about how we design the BCI mapping and how we change the mapping. So a typical BCI mapping looks like this, where we have neural activity. So typically we're recording from 90 channels of activity. So it's 90-dimensional. We pass it through a Kalman filter, and the Kalman filter produces two-dimensional cursor velocities, horizontal and vertical. Okay, so what we did was we introduced factor analysis as a first stage here, okay, whereby we use factor analysis to identify 10 factors that represent the axes of the intrinsic manifold. Okay, and this number 10 was determined using cross-validation on the data. Okay, and what this allowed us to do was to define these two different types of mapping changes. So in the cartoons that I've been showing you, we've been looking at 3 neurons, a 2-dimensional intrinsic manifold, and a 1-dimensional BCI mapping. Okay? In our actual experiments, we had 90-dimensional neural activity, roughly. We had a 10-dimensional intrinsic manifold and 2-dimensional cursor velocity. Okay, so to define a within-manifold perturbation, okay, what we did was we took those 10 factors that came out of factor analysis, and we permuted its elements. Okay? So now the definition of the factors is the same, but the relationship between each of these factors and movement is changed. Okay. In the second type of perturbation, for the outside manifold perturbations, what we did was we permuted the order of the neural units, okay, before we input them into factor analysis. Okay, so say 2 neurons, which positively correlated before, may now need to negatively correlate. Okay, so in terms of experimental methods, we used Utah arrays implanted in the primary motor cortex of macaque monkeys. Okay, we recorded threshold crossings, so we didn't do spike sorting here. And we took spike counts in non-overlapping 45-millisecond windows, and the monkey performed an 8-target center-out task. Okay, I'm going to show you a couple representative sessions. So here is an example of a within-manifold perturbation session. So at the beginning of the session, the animal uses this intuitive mapping, which it can control very well. Okay, so its performance is usually at 100%. At some point, after a few hundred trials, unannounced to the animal, we switch the mapping. Okay, and then, as you would expect, performance goes down. Okay, and as the animal experiences errors, right, it can learn, right, and then we see that the performance recovers. Okay, and then at the end, we show the intuitive mapping again during a washout session. Okay, so contrast this with an example outside manifold perturbation. Okay, so here again, good performance at the beginning. Okay, we then introduce this perturbed mapping unannounced to the animal. Performance goes down, and we see that performance does not recover so well. Okay? So the key takeaway from this slide is we see lots of learning here and little learning here. Okay? So the key result, okay, is that we see greater learning for within manifold perturbations than outside manifold perturbations. Okay, so to summarize this part of the talk, okay, what we found was that we saw more learning within the intrinsic manifold than outside the intrinsic manifold. Okay? And so this implies that the existing structure of a network can shape learning. Okay. We use dimensionality reduction in this study, okay, and there have been many studies that have used dimensionality reduction to visualize population activity and to create hypotheses. Okay? And in this study, we used dimensionality reduction and took a step further, okay, by incorporating dimensionality reduction into the experimental design to reveal causal constraints on the activity patterns that a population of neurons is able to produce. Okay. So now I'd like to turn to the second part of this talk, okay, which is to ask, how does neural population activity change during learning? Okay, so what we're going to do is to focus on those sessions in which we saw the most learning. Okay, and these are the within manifold perturbation sessions. Okay, and then what we're going to do is to zoom in to and look down onto the intrinsic manifold, okay, and ask how neural activity is changing, okay, within the intrinsic manifold. Okay, so here's the picture that we have before learning. Okay, so we have this BCI mapping here, right, and this is a BCI mapping that the animal can control very well. Okay, so these green dots correspond to activity when the animal wants to move the cursor to the left. Okay, and these purple dots represent times when the animal wants to move the cursor to the right. Okay, and you see there's a good match between the neural activity and where the BCI mapping maps the cursor to move to. Okay, now we switch the mapping. Okay, and these neural activity patterns are not going to work as well for controlling this perturbed mapping. Okay, so our key question here is, how does the neural population activity reorganize within the intrinsic manifold, okay, during learning? Okay, so our approach is to first form hypotheses of how the population activity might change during learning. And then we're going to predict what the after-learning population activity patterns look like under each hypothesis. And then we're going to compare these predictions with data. Okay, so our first hypothesis is called realignment. Okay, so realignment seeks to maximize behavioral performance. Okay, so what you see in this picture here is that the clouds of activity move in a way such that now, whenever the animal is intending to move the cursor left, neural activity is now sort of up here in the right place. And when the animal intends to move right, neural activity is down here. Okay? This would control the cursor really well under this perturbed mapping. Okay, hypothesis 2, okay, is called rescaling. Okay, so rescaling tries to restore the influence of activity on behavior. Okay, so what I mean by this is under the intuitive mapping, the neural activity along the horizontal axis here has a large influence on behavior because the intuitive mapping was sort of horizontal. Now, after the mapping switch, movement horizontally no longer has a large influence on cursor movements. And so this hypothesis says that perhaps the variance of the activity would scale, right, to restore the influence of horizontal movements of the neural activity on cursor movements. Okay? And similarly, along this vertical axis, the influence increases, and so we would scale the variance of that activity down. Okay? And this is an analogy to visual motor gain adaptation with arm movements. Okay? But here, it's in the neural activity space. Okay? Our third hypothesis is called reassociation. Okay? So in reassociation, we hypothesize that neural activity is repurposed, okay? Meaning that the same cloud of neural activity is here, as we saw before learning, but now we recolor the dots. Okay? So basically, the animal might use the same neural activity patterns, but now for different movements. Okay? So all of these hypotheses predict after learning neural activity that is within the intrinsic manifold. Okay? Also, they respect the physiological limits on firing rates of individual units, right? Because if some of these hypotheses predicted unrealistic firing rates, right, that would not be an interesting test here. And then thirdly, all of these hypotheses predict realistic neural variability for each movement direction. Okay, so the trial-to-trial variability. Okay, so when we started asking which of these hypotheses is a good match with the data, we visualized the population activity by projecting them into different spaces. Okay, and the striking effect that we saw was that the cloud of activity before learning was exceedingly similar to the cloud of activity after learning, no matter which projection we looked at. Okay. So this was a clue, right? So now we want to quantify this. Okay, so we used two main quantifications. So the first is repertoire change. Okay. So that's what's plotted along the vertical axis here. Okay, 0, okay, means that the cloud is rock solid. Okay? A positive repertoire change, okay, means that the cloud either shifted or expanded. And a negative repertoire change means that the cloud contracted. Okay. So what did each of these hypotheses predict? Okay, so realignment and rescaling both predict that the cloud moves or gets larger, whereas reassociation predicts no change, right, in the repertoire, okay, as we would expect. Okay, we compared this to the data, okay, and the data also showed no repertoire change. Okay, so our data is consistent with reassociation according to this metric. Okay. The second way in which we quantified this was by change of variance, right? So how much variance was there in the population activity along different dimensions? Okay. So the horizontal axis here, what we're going to plot is a change in variance of the activity, pre-learning versus post-learning, okay, along the intuitive mapping. Okay, whereas the vertical axis here is a change in variance of the neural activity, pre-learning versus post-learning, according to the perturbed mapping. Okay? So this space is going to allow us to distinguish between these three hypotheses and compare to data. Okay. So for realignment, what we see is that there is an increase in variance of the neural activity along the perturbed mapping. Okay? So that prediction is reflected here in these points, right? So each of these data points corresponds to one monkey. Okay? So this is the prediction based on the neural activity from that monkey. Okay? So we see a large increase in variance along the perturbed mapping and little change along the intuitive mapping. In contrast, for rescaling, we see an increase in variance along the intuitive mapping, okay, and little change along the perturbed mapping. So that's why we get points here. And then reassociation, okay, predicts little to no change, right, in the variance along either of the two mappings. Okay? And so that is why the blue points are here near 0. Okay. The data are shown here in black. Okay? And the data are also here near 0. Okay? Which is a good match with reassociation. Okay? So there is little change in the variance of the neural activity, okay, pre-learning versus post-learning. Okay. So one other test that we did in comparing these different hypotheses, okay, was to ask whether they are predictive of behavior. Okay. So something that we observed consistently in these experiments was that learning was imperfect. Okay, so what do I mean by this? Okay, so on the vertical axis here, we're measuring acquisition time. Okay, so this is the time it takes for the animal to move the cursor from the center of the screen to the target. So under the intuitive mapping, the acquisition time is about 1 second. After we switched the mapping, the acquisition time went up. Okay, the monkey did not perform as well. And then over the course of learning, the acquisition time would get better. Okay? But something that we consistently observed was that the acquisition time did not come back to its original baseline level, okay, shown by this gap here. Okay? And what we asked was whether these different hypotheses would predict that. Okay. So here are the predictions for each of the hypotheses. Okay? So realignment, it turns out, predicts acquisition times that are even better, not only better than what the animal showed after learning, but even better, oftentimes, than what the animal showed under the intuitive mapping. Okay. Rescaling, okay, often predicted acquisition times that was a little bit better than what the animal showed post-learning. And then reassociation did a good job at predicting what the animal's performance would be after learning. Okay, so these 3 lines of evidence, okay, suggest that reassociation is doing a good job at predicting what neural activity looks like after learning. So it appears to us that the neural activity is constrained to lie within a repertoire, okay, whereby when the animal is learning, rather than showing new neural activity patterns outside of this repertoire, what the animal is constrained to do is to reassociate or repurpose the same neural activity patterns to produce different movements. Okay. So in the first part, right, we showed that learning is constrained by an intrinsic manifold. Okay? And here in the second part, we show that there is yet a further constraint, okay, within the intrinsic manifold. Okay? That it appears as if within the intrinsic manifold, the population activity is further constrained by this neural repertoire, okay, during learning. Secondly, what we found was that learning is suboptimal, right, on the time scale of hours, right? And that the fact that the neural activity is restricted to this repertoire might explain why learning is suboptimal on this time scale. Okay, in the next talk by Emily Obi, okay, what we're going to show is that we can ask the animal to learn over multiple days. Okay, and it turns out that these constraints can be overcome, okay, with long-term learning. Okay, so now I'd like to sort of take a step back, okay, and come back to the theme of this talk, right, that I introduced at the very beginning. Okay, and ask, you know, what did BCI bias, right, in this experiment? Okay, so this experiment that would have been very difficult to do without a BCI, right? Because with a BCI, we were able to place the task constraints directly on the neurons themselves, okay, rather than indirectly through requesting particular arm or eye movements. Okay. Secondly, BCIs enabled this work because we can causally attribute behavioral changes, i.e. changes in the cursor movements, to specific changes in the activity of the neurons that we observed. Right? This is typically not possible when studying eye or arm movements because we typically don't know the causal relationship between the neurons that we record and behavior. Thirdly, right, in the BCI, we define the neuron to movement mapping, and we could define it to be linear. Okay? And this was enabling for defining the various hypotheses in the second part of this talk. Okay, I'll also mention here, without going into any details, that we're also using BCI to study internal models of sensory motor control, okay, as well as neural redundancy. Okay, so taken together, okay, what I've hopefully convinced you of, okay, at least as a starting point, is that BCIs represent a powerful tool for basic science. And the reason for this is that BCIs simplify the brain's output interface without hopefully simplifying the complexity of brain processing that we seek to understand. And I think through ongoing work in our groups as well as other groups in the field, what we're starting to see is that BCIs engage many of the same cognitive processes, okay, as in arm reaching or eye movements. Okay? In particular, questions about learning, internal models, and more. Okay? And even more broadly, BCIs could perhaps be used in other systems, right, outside of M1, to study sensory, motor, and cognitive processing. Okay, so I'd like to acknowledge those who contributed to this work. These are people highlighted in red here, as well as our funding sources. Thanks.
Thanks to Byron's impeccable timing, we have plenty of time for questions.
Do you believe that being in motor area do you believe that being in primary motor area has a big consequence on your on particular hypothesis, which was true or not?
Yeah, I think this is a really interesting question, and I think it would be interesting to do a similar experiment in other brain areas adapted to those other brain areas.
Yeah, thank you for a beautiful result. So when you pick a new 2D space randomly in the 2D intrinsic space, maybe that is almost orthogonal to the previous space. But if you choose the new mapping within the original 2D space by flipping or rotation, can learning become easier or more difficult?
Yeah, we didn't test that explicitly, but yeah, those would be interesting hypotheses and would be analogous to visual motor rotations.
Right. Yeah.
Yeah, we didn't do those experiments, but I think this was sort of part of the design of our experiment, in that the perturbations that we did were in this 10-dimensional manifold, right? And so they're not simple sort of 2D visual motor rotations where the animal can simply solve it by, say, rotating where it's aiming to move the cursor or sort of flipping left and right.
Right, yeah. Thank you.
Yeah, sorry.
So, yeah, how much do you think that the constraints on learning that you have observed are due to the locality or the low number of neurons that you have measured? Like, presumably they're constraints because you are measuring a very small patch of the tissue.
Yeah, I'm not sure, sort of a priori, whether recording, how the number of neurons that we record would affect the ease of learning in this task. Because I think one could come up with hypotheses in either direction about which would make learning easier or harder. I don't know if you have any specific thoughts.
No. I meant that, so the reassociation constraint, right? That you don't see like major realignments of the learning direction. That could be because neurons in a local patch are highly correlated.
Got it. Yes. So I think it would be really interesting to record from other brain areas during this task, right? To see whether reassociation also happens in other brain areas, right? I would hypothesize that in some other brain area, right, I would hypothesize that we don't see reassociation in all areas, right? Because in some brain area, there has to be a change in, for a particular desired movement, a different neural activity pattern has to be produced, right? And so maybe something like realignment would happen in a different brain area. So my short answer would be, yes, it probably depends on the population of neurons that we're recording from, but to be tested.
So in the first part, I noticed that when you did the outside manifold perturbations, at least in the example graph you showed, the success rate didn't seem to go down to 0. It only went partly down. Is that something that is consistent across other experiments? And what do you have to say about the...
Yeah, thanks a lot for pointing that out. So a really, really important control in this experiment is that the initial difficulties were matched for the two types of perturbations. So what I showed you were 2 example sessions, and unfortunately, the initial difficulties were not matched for these particular 2. But across our tens of sessions that we ran, the distribution of initial difficulties was indistinguishable. Okay? And this is very important for this experiment because we want the starting point of learning to be the same for the 2 types of perturbation, and then see how the animal can learn afterwards.
Thank you.
Yeah, sorry.
So 2 quick questions about interpreting the intrinsic manifold thing. So one might be related to the answer to that question, which is, so in 90 dimensions, 2 vectors are going to be much more orthogonal than in 10 dimensions when chosen randomly. So do you control for that? Do you choose out of intrinsic manifold perturbations to match the angle to your previous, to the intrinsic, to the intuitive mapping?
Right.
The same statistics as within the 10D perturbation?
Yeah. So there are many, many possible outside manifold perturbations. Right? And so the way in which we chose these outside manifold perturbations was during the experiment, right after this intuitive mapping, we would enumerate millions of different possible outside manifold perturbations, and then predict for each one how difficult it would be for the animal initially to use that mapping using the neural activity we had just recorded. Okay, and the reason why we did that prediction for each of the millions of outside manifold mappings was that we wanted to choose an outside manifold mapping of intermediate difficulty. Okay, so if the outside manifold perturbation were too hard, the animal would give up and not learn. If the perturbation were too easy, that would not really encourage learning, and so that would not be interesting. Right? And so, so we use that procedure to select which of the many possible outside manifold mappings we would use on any particular day. But I don't know if your question was sort of even deeper about sort of how that affects the amount of learning that we see.
Yeah, I guess it was just basically saying if you're, say within, going back a couple questions, within the 10D manifold, let's say you just rotated the vector by a couple degrees. Presumably that would be, they would learn faster. And so okay, one thing you can do instead of asking within intrinsic manifold without is to take the two vectors from intuitive mapping and the new one and just say as a function of the angle between those vectors, how fast is learning or how successful is it by the end of the session. And because certainly choosing randomly, you would have much higher angles on average and outside the intrinsic manifold than within it. To the extent that there is something that big angles are hard to learn, that could trivially explain the answer. Of course, since you're doing this pre-selection process, maybe you've eliminated that effect already because maybe you're choosing a set of outside manifold things that have the same angle distribution.
Right. So a really important part of this experiment was to make sure we were doing an apples-to-apples comparison. Okay. So in addition to matching the initial difficulty after the mapping switch, what we also did was to match the angles by which we rotated the mappings in both of them. And furthermore, we matched the amount of preferred direction changes that were required for these neurons to produce under these two different types of mappings. Okay. And so comparing these two different types of mappings, the main thing that distinguished between these two types of mappings was whether the mapping was entirely within the intrinsic manifold or outside of it.
Thank you, Byron. We're going to move on to the next speaker.